Welcome to week, what are we on, 13 now? Yes, 13. So just a few more weeks left. This week I decided to cover chapter 26. This is just an overview of defenses against disease. I find this chapter really interesting. I feel like I say that in every presentation. Like, I like this fact because, but alas, it is true. I do think it's interesting to understand how the body works and just on the level that I think people don't talk about in everyday conversation. I hope you all survived your exam week well. So let's get started. This is the overview of the immune system. So we have the immune system. You already know fights infections and cancer. It's what keeps you healthy. Then you have your lymphatic organs. That's your red bone marrow, your thymus gland, your lymph nodes, and your spleen, tonsils, and your appendix, and then those are patches of immune tissue, so pieces of them, and they work to help keep you healthy. This is a picture representation. The picture for this is on page 505, and you can take a closer look. It shows you where your lymph nodes are, your whole like a lymphatic system, and where the lymphatic organs are, and then a breakaway view of what an actual lymphatic organ looks like, or a lymph node, which I think is interesting because these are things that like you know of, and it's sort of this abstract way of thinking. You know that everybody has them and they help keep you healthy, but to have a visual to think of how they're designed in the body, I think kind of just adds to it. So the lymphatic organs. We have red bone marrow. Those produce blood cells. In children, most bones have red bone marrow. In adults, that is reduced to just our skull, our sternum, our ribs, clavicle, our vertebral column, and then the ends of our humerus bones and the femur, which like the nearest part attached to the body. Things change a lot between childhood and adulthood, as you know, you probably already know that. And for those of you that do know this information, wonderful. And those of you who don't, that's also fine. We're here to learn together. So the lymphocytes, they can differentiate into either B lymphocytes, which are B cells. Those complete their maturation in bone marrow, and they produce antibodies. And then you have your T lymphocytes, or your T cells, and those complete maturation in the thymus, and they kill antigen-bearing cells outright. And you've probably heard of T cells before in reference to HIV, but B cells maybe not so commonly. We'll go over all these. So the thymus is larger in children. Again, going from childhood to adulthood, things change. So that's going to shrink as we age. But it plays a role in how the T cells mature. So only about 5% of T cells ever leave the thymus. If any react with the self, then they die. So then your lymph nodes, those are small ovoid structures that occur along the lymphatic vessels. And you can see that in the picture on page 505. Kind of breaks down the way that they look. They're kind of like little dots, little knots along the stretch of lymphatic vessel. Those filter the lymph and they keep it free of pathogens and antigens. So they're kind of like the net. They catch things that aren't supposed to pass through the rest of the lymphatic vessels. I have a feeling I'm going to really tongue tie up these words, so forgive me if I do that. I don't know what's going on today, but the L's and the PH's and the V's are just too close together, and I just feel like it's gonna happen. So anyway, the nodes will filter out a lot of the bad stuff. Then your sinuses are lined by macrophages that engulf and destroy pathogens. You also have this instrumental thing it's involved in fighting infections and cancer due to many of the lymphocytes. Swollen lymph nodes may indicate that the body is fighting an infection. So I think this is probably where a lot of people may be familiar with lymph nodes. They feel around, their neck, or maybe like the armpit section of the body, and they're like, man, it feels like there are little like knots in there. And so if you're sick and you're paying attention, you have awareness, self-awareness, um, you will notice that. And sometimes when you go to the doctor, they'll check for swollen lymph nodes too if you're not feeling well, and that's because they're catching the things that are making you sick. They're working on it, and they kind of like swell up because they're fighting away in there. So then you have your spleen that filters the blood. Red pulp, this is blood passing through the sinuses that is filtered of pathogens, debris, and like other worn out blood cells. And the sinuses are lined with the macrophages. I mentioned that a second ago. Then you have the white pulp. Lymphocytes, those are actively engaged in fighting infections and cancer. Like we talked about, they swell up because they're actively engaged. The white pulp, it can survive without a spleen, but then without the spleen to kind of protect it, they're more susceptible to infections. So I said that backwards. We can. I knew it was going to happen. We can survive without a spleen, but it means that we'll get sick easier. Goodness. We have a variety of immune system cells. So immunity, by definition, is the body's capability to repel foreign substances, pathogens, and cancer cells. You have non-specific immunity, that's indiscriminate, and then you have specific immunity, that is a very specific antigen. And an antigen is any molecule that stimulates the immune system. Here we have just a brief list of some of the cells that are in the immune system and their functions. I won't go over every single one of these, but you can find that same chart on page 506. You can read through each of them. You have your macrophages and what they do, your mast cells, neutrophils, natural killer cells, your B lymphocytes, plasma cells, memory cells, all of that. Read through each of these, pause this if you want to, or turn to the page in your book. But basically, there's just a lot of different cells. They all have different functions in different areas of the body. So non-specific diseases and innate immunity. That's the first line of defense. You have barriers to entry. You have an inflammatory response, complement system, and natural killer cells. The inflammatory response can be quite frustrating, just depending on the wound. Sometimes you get inflammation without doing very much. So that's part of it. So the barriers to entry, you have mechanical barriers to entry, skin, obviously, your mucous membranes that line your respiratory, digestive, reproductive, and urinary tracts. And then you have oil gland secretions that contain chemicals that'll weaken or even kill like certain bacteria on the skin. So these are things that may sound familiar. You know the structure of the skin, so you have the cells of the epidermis. They'll harden and die as they progress to the outer layer of the skin. The outer layers of dead cells, they form a protective barrier against the invasion, and then sweat and oil glands are acidic enough to inhibit invasion by bacteria. The upper respiratory tract, that is lined with what they call ciliated cells. They sweep mucus and other like dirt and trapped particles up and out of airways. The stomach, we know, is full of acid, acidic pH to inhibit or kill bacteria. And then the resident bacteria in the intestine, the vagina, other areas, those also prevent pathogens from multiplying. And we've talked about, I think it was early in the first few weeks, we've talked about the healthy bacteria. That is what this is referring to whenever we speak of resident bacteria, the bacteria that we hold in our bodies to help keep things in balance. The inflammatory response, that is a series of events that occur whenever tissue is damaged. So you have your inflamed area that has 4 outward signs. You have redness, heat, swelling, and pain. I think most of us have experienced this at some point in our lives, and if you haven't, then you're extremely lucky and let's trade places. Mast cells release chemicals, So you have histamine that causes the capillaries to dilate, they become more permeable. You have excess blood flow that causes the skin that'll redden and warm your skin, make it really flushed. Then you have the increased permeability that allows fluid to escape, that causes swelling. And then you have the swollen area that stimulates nerve endings, resulting in pain. So you may have one or all of these depending on the degree of an injury. I've experienced all of these, not all at the same time always, but sometimes all at the same time. Like I said, it just depends on what's going on. One small example that comes to mind is whenever I would walk for 15 miles a day, which sounds excessive. It's something like 40 something thousand steps over different kinds of terrain. Sometimes it was rock, sometimes it was like mountainous, sometimes it was sandy, sometimes it was muddy. But whenever I first started doing that, for a job, of course, I didn't have any sort of what would you call that? Conditioning. I didn't have any conditioning built up. So of course, after a few days of that, you get blisters all over your feet. Even if you have the proper shoes, you just have to build up the conditioning in the body for that. So at first, I can tell whenever I was going to get a blister on like one side of my heel or the other because it would itch. You have this histamine response, and I could tell that something was happening under there. Sometimes the area will become red, not always. But then, you get the swelling and the fluid in the actual blister itself. And then as the blister, sometimes if you pop it, this might be a little gross and I apologize for that. It's just an example. Sometimes there's pain around the area, especially if you're still walking and because you can't really not use your feet. So that's just a small example. If you've ever had a blister anywhere, just from like overuse or a burn even, you can tell that's an inflammatory response. That's my whole point here. Then you have neutrophils. Those are phagocytic white blood cells. So those migrate to the site of the injury. They get sent there to handle business. And that encloses the pathogens and vesicles for digestion inside the cell. So they are the army that gets sent out to capture the pathogens and handle it. This is again a picture that shows you the inflammatory response. So it is on page 508. I think it's very interesting. I like the pictures because for me, I'm a visual and hands-on learner. I can read something and get it, but it really helps me if I can read it and write it and see it. It really helps things sink in. So if you would like, just take a moment to look at the picture on 508 or pause this here and look. It just shows you the barriers to entry, your protective proteins, the cells, the way the inflammatory response works, and kind of like just arrows and labels for things. And you don't need to know every single thing in this picture, but just go over it for your understanding. And then we have the complement system. The complement system, that is usually just called complement. That's composed of different blood plasma proteins designated by the letter C in a subscript. It's like a domino effect. So you have an activation of one will stimulate more. So and that's kind of like what complement is referring to. So you start it and it's just this long chain of events that keep happening. So complement certain immune responses. So that's what this means. Involved in and they amplify inflammatory response by attracting phagocytes. Some will bind to the surfaces coated with the antibodies to ensure phagocytosis by a neutrophil or macrophage. Now, as a reminder, phagocytosis, that's the ingestion of bacteria or other materials whenever they go and they capture the pathogens. The complement system is, like I was saying, is continued. So the blood plasma proteins, they complement certain immune responses. They can release growth factors that cause the red bone marrow to produce or release more white blood cells. primarily neutrophils. So things that are going to just enhance the response of whatever's happening. The macrophages, they'll fight infection. They travel to the lymph nodes to activate the lymphocytes to help the fight. So they're recruiting. And then pus is actually dead neutrophils, cells, bacteria, and living white blood cells. So while pus is gross, I mean, it usually just means that healing is happening. The ways to get rid of pathogens. So the complement can also form this membrane attack complex that will then insert itself into the plasma membrane and helps like pop the invader. So it essentially just like pokes a hole in the cell and blows it up, which I think is really cool. Like, it's a cool way to think about it. And then you have your natural killer cells. These are the cells that are they're not recognized as self. I feel like that is self-explanatory, but just in case. So anything that is not a non-self antigen, something that comes from outside of the body. So these natural killer cells will attack things that they don't recognize as yours. And they use the same methods as T cells do to kill cells. They're not specific, so they don't increase in numbers when they're exposed to a specific antigen. There's no memory or previous contact. So, responses to antigen. So an antigen, that can be part of a pathogen, a foreign cell, or a cancer cell. We become immune when our body has learned to destroy a particular antigen, a specific one, that distinguishes self from non-self. Our immune system does not respond to the molecules on the surface of our own cells. Lymphocytes have a plasma membrane and a receptor for antigens like a lock and key. We have lymphocytes for any possible type of antigen. Adaptive immunity usually lasts for some time, so it's a shorter amount of time. Once we recover from like the measles, we don't usually get it again. That is due to the B cells and the T cells. So B cells mature in bone marrow. That gives plasma cells the ability to produce antibodies. And then the T cells mature in the thymus gland. That those don't produce antibodies. Some of them can regulate immune response, but others directly kill cells bearing antigens. So they function differently. The B cell response is when each B cell can only bind to a specific antigen. The antigen that fits its receptor binding site, or the BCR, the B cell receptor. Some B cells never do a thing. They just are useless. No antigen ever binds to its receptors. What I mean by that. So if an antigen does bind to the BCR, that particular B cell is activated, it divides, and then it starts producing plasma cells and memory cells, and that is stimulated by secretions called cytokines. Cytokines? Oh, I forgot how to say that. Hold on. Had to double check myself. Cytokines. Okay, that's right. Just to put in perspective, I'm from a very southern part of Arkansas, so there's a lot of words that I said wrong for a lot of years. So I just am a little paranoid, and occasionally you're going to hear me say something really weird, and just, let's just move on from those things when they happen. So characteristics of B cells. They provide an antibody response to pathogen. They're produced and become mature in bone marrow. They live in the lymph nodes and the spleen. They circulate in the blood and the lymph. And they directly recognize antigens, and they can undergo cell division. And cell division produces the antibody secreting plasma cells as well as memory B cells. So this is your overview of the characteristics of B cells. Again, another picture. I like these. You can clearly see what happens and where they move. Antigens and pathogens, cytokines are all clearly marked. They attach. You can pause and take a look at this if you would like. It is also on page 510. the B cells and the antibody response. So plasma cells are pretty big with a extensive rough endoplasmic reticulum for making and secreting the antibodies specific to the antigen. So they're large and in charge. And then memory B cells, these are responsible for our long-term immunity. This is the reason why when we get vaccinated for certain things, we are immune for life. We don't have to keep going back and keep going back. That's not the case with everything, but with a lot of them. If the same antigen enters the system again, the memory B cells will divide and quickly produce what they need to pump out those antibodies and get rid of it. Once the threat passes the development of the new plasma cells, that stops, they quit, and then those undergo apoptosis. That means cell death, a programmed cell death, apoptosis. The function of antibodies. So you have something called immunoglobulin proteins that are capable of combining with a specific antigen. Antigen, that's like I said before, an antibody reaction that can take several forms. So anything that causes an antibody reaction, that's an antigen. Those can produce complexes of antigens combined with antibodies or immune complexes that mark antigens for destruction. So the antibodies come in and they tag all these antigens and they're like, these need to be destroyed. Here's the overview of the structure of an antibody on page 511. They're not complicated. That, for as simple as they are, they do a lot of work. Blood antibodies, they're a little different. So you have letters that stand for antigens on your red blood cells. This is how we have, ABO blood labels. If you have type O blood, you don't have either A or B, obviously. Type O has both anti-A and anti-B antibodies. You cannot give a person with type O blood a transfusion of type A blood. Their anti-A antibodies, that's a tongue twister, will combine with the type A blood. And you'll get something called agglutination, which is a clotting or a clumping of red blood cells that causes them to burst. There are other antigens in addition to A and B that have to be considered for transfusions. So it really freaks me out to think about how they learned this before they realized about blood types, back before modern medicine. But I'm glad that they figured it out. It gets a little complicated. And you may have heard before, but so anybody can take type O blood. If you have, A or B, you could take type O. But type O can only take type O because of these specific anti-A and anti-B antibodies that are in the type O blood. It just kind of like rejects anything else. So here's a list of the blood types. You have A and B, AB, and O, of course. And then I think you'll have a positive and a negative of each. So I personally am A positive. I went to a blood drive one time and donated blood. I've done it several times since, but the first time they let you know what your blood type is, you get like your certificate saying you gave blood, and that is how I learned. And in some places, I believe that blood type is similar to the way that people think about horoscopes, like, oh, you're a Virgo, you have these traits. Some people will be like, well, what's your blood type? And that has like certain personality traits. It's been a while since I've looked that up, but I just find it kind of intriguing because it's not something that happens here in the United States, at least not that I know of. So moving on. T cell response. So when T cells leave the thymus, they have unique T cell receptors or TCRs. Unlike B cells, the T cells, they can't recognize an antigen unless they have help. So they have to have somebody tell them, pay attention to that. The antigen has to be presented to them by an antigen-presenting cell or an APC. So after a macrophage phagocytizes a virus, then it's digested in a lysosome. So it's encapsulated, and contained. And the antigen from the virus is then combined with a MHC, or like a self protein, something that the body can recognize, and then move to the surface of the cell for presentation to a T cell. So I know there's a lot of like fancy words in there, but all it's really saying is the macrophage goes in, it encapsulates it, and then the antigen from the virus combines with the protein that can be recognized by the cell, and that's slapped to the outside of the cell. So then a T cell is like, oh, it's like a headlight. It's like turns it on, and the T cell is like, oh, there you are. I see you now. The T cell then is activated and divides to produce more T cells, either cytotoxic or helper. And then as the infection wanes or goes away, the T cells undergo apoptosis. So it just has to have a little help figuring out where to go. And then they kind of do their thing from there. And then as the infection goes away, then they're just like, okay, we're done. So some of the characteristics of T cells, they provide a cellular response to virus-infected cells and cancer cells. They're produced in the bone marrow, and they mature in the thymus. An antigen, like I just was talking about, they have to be presented to a T cell in a groove of an MHC protein, so that self-recognizing tag. Cytotoxic T cells, they destroy those non-self antigen bearing cells, so something that is outside that is not supposed to be there. And then the helper T cells, those are the ones that secrete the cytokines that control the immune response. There's the overview of the activation of a T cell. That one is on page 512. And then the T cell response. So here's some more. These are the separate T cells I was just talking about. So for the cytotoxic T cells, they store vacuoles that contain something called perforins. Those are what can puncture the cell membrane. Or granzymes, those are delivered into the pore to cause apoptosis. So one or the other. And then they're responsible for responses to virus-infected and cancer cells. And then you have the helper T cells. Those help regulate immunity by secreting the cytokines. They signal chemicals that stimulate various immune cells to perform their functions. So all the things we've been talking about, macrophages, B cells, and other T cells. And then, so this is what happens here. HIV actually infects the helper T cells, making the AIDS patient susceptible to infections. So the things that are kicking your immune system into gear and helping fight infection are hijacked and attacked pretty much, and then your immune system lowers, so then you become more susceptible to just different diseases, anything. Your immune system lowers and it can no longer fight the way that it needs to because the fighting system has been taken over. I actually think this picture is really neat. Just the electron microscope pictures blow my mind. That's what's on the left, obviously. Just to kind of show you what it actually looks like. The pictures are cool. The artistic renditions are very cool, but I love to see the real thing. That's on page 513, if you want to look at the breakdown. Now we talk about tissue rejection. So tissue transplants and immune response. Certain organs could be transplanted pretty easily from one person to another if the body doesn't reject them. But rejection occurs because those cytotoxic T cells and the antibodies bring about the destruction of the foreign tissues in the body. It's not recognized as self, and so then it is foreign, and the body decides it needs to get rid of it. It's a danger to the working system. Organ rejection can be controlled by carefully selecting the donor, first of all, but then also administering the immunosuppressive drugs. So it's best if the donor organ comes from a person with the same type of the MHC proteins. And some of the drugs work by inhibiting the response of the T cells to cytokines to weaken the immune response, so then the body can kind of accept the new organ. Now, this is not a temporary thing. Whenever you have to take the immunosuppressing drugs to allow your body to accept the organ, you have to stay on those for the rest of your life. And then it does lower your immune response, so it can make it hard to heal from any sort of sickness. All right, now we've reached the part of the chapter that talks about immunizations. And because of our immune system and the way that things are set up, that is why vaccines work the way that they do, because they figured out the system. So vaccines, of course, by definitions, are substances that usually don't cause illness, but cause the immune system to respond to them, and you can build up a memory. So pathogens or their products, typically, that have been treated so that they're not virulent are used for vaccines, and the proteins produced by genetically engineered bacteria. You can get active immunity or passive immunity. And I don't know how much you know about vaccines, but I like to, just the easy way to remember it are like live versus dead vaccines. So they'll either use a live form of the virus, which typically gives you a longer immunity time, or what they consider an inactivated or a killed version of a germ that causes a disease is used to make a vaccine. And with those, you have a shorter immunity protection time, so it's not always as strong as, say, like a live vaccine, and you have to get things like boosters. And so, but there are newer technologies, like those that have been used with COVID-19, are using messenger RNA or mRNA molecules to produce specific parts of the virus to act as antigens. So the cool thing about science is that it's never really cut and dry, and just because something has worked for a long time doesn't mean that there may not be a better way. Scientists are always hungry for this type of discovery. That's the driving force behind a lot of scientists. We have questions. What if this? What if that? Maybe I could try it like this. Would this be better? And we also have to be open to new ideas. Just because this has worked, it doesn't mean, okay, we're done. We'll stop here. I couldn't imagine the state of the world if people just stopped when something worked instead of trying to find a better, more efficient way. But yeah. Here is an overview of the mRNA vaccines using the mRNA of the S protein of SARS-CoV-2. COVID-19. So researchers were able to develop this new method of producing vaccines in response to the pandemic. And S is a spike protein. The S protein is a spike protein. And it's called that because on the outside of a cell, they look like a bunch of little spikes. So in order to make an effective vaccine, the researchers identified that spike protein, and then an artificial mRNA sequence for that protein was made and placed in a lipid coating to use as this vector for delivering the mRNA in the vaccine. So it's its delivery method. And then once that's injected into the body, that mRNA is translated into millions of copies of that S protein, and then the S protein invokes the immune response from the body. So they have synthetically made this response by IDing and honing in on that spike protein, and then they figured out a way to make this work. And RNA in general is sometimes difficult to work with. So this is, I think, a pretty big leap in vaccine making for sure. Active immunity is promoted by immunization. So after that vaccine is given, the antibody titer in the blood can be measured. A primary response is the first response to this antigen. Obviously, primary means first. The secondary response is faster and then produces more antibodies. That would be like the booster dose of the vaccine. So you have your first response, and then as you get your booster, your system responds faster because it already kind of recognizes things. Active immunity depends on the presence of memory B cells and memory T cells. And they usually are long lasting, but a booster might be required after several years. And if you've ever looked at your vaccination schedule, or if you have children and you've had to take them for their MMR and then like a booster, you know all about this process. When they're little, it almost seems like it's never ending, as you have doctor's appointments all the time. Oh, you're due for a vaccine. Active immunity due to immunization. This is just a chart that kind of puts into perspective your primary versus secondary response. So the visualization, so you have your first, you get a little curve down here at the bottom, and then as you get your secondary, your booster, your system can really kick it up a notch. So I mean, it's a pretty big difference. I like charts. I like statistics, because you really I find it very difficult to argue with legitimate statistics, and then I think that's the point. Now, passive immunity is prepared antibodies or immunoglobulins that are not produced by the individual's plasma cells. They're temporary. They don't have a memory. They're like a fair weather friend. They're only there for a little bit, and then before you know it, they're gone. And who knows where they went. Newborns receive antibodies from their mother across the placenta or through breastfeeding, but they only last a few months. So a lot of times that's why they say that breastfeeding babies are best, because you get those antibodies from the mother that keeps babies healthy for as long as they're breastfeeding. And then if you have children, it seems like they're sick all the time because they're building their immune systems. But early in the first few months as they're growing, they need all the antibodies they can get, so they can slowly build their immune system. Injections of what they call gamma globulins, or portions of blood that contain antibodies, are given to patients who are like unexpectedly exposed to an infectious disease. So it's a little boost of antibodies to make sure that they don't get sick or the disease. There are disorders of the immune system. So the immune system cannot distinguish self from non-self, which we've gone over several times. But something like allergies, hello, it's me. I have those all the time. I think a lot of people do. I am not alone in my struggles. Hypersensitivities to substances that ordinarily would not cause an immune response, like pollen, food, animal hair, etc., etc. An allergen is the antigens in an allergy. I am so when I'm an asthmatic, right? So when I was little, I was really, really bad. They were trying to figure out what my triggers were, so they did a lot of allergy tests on me, and I'm allergic to a lot of things. But the worst things, cats are number one, which is extremely sad because I love them and they love me. I just can't be around them. Definitely can't touch them and then touch my face or anything. I'll end up in the hospital. Pollen definitely aggravates me. Whenever I worked out at walking the wind turbines, looking for post-mortality surveys on construction sites, they had a lot of cornfields. They would harvest the corn, and the corn dust was killer. It really, really messed with me. I don't think I've taken so much allergy medicine in my life until that point. I actually ended up with a sinus infection. I think I told you guys about that earlier in the semester. But yeah, so pollen, food, animal hair, dust, that sort of thing. When I was little, laughing, if I did too much laughing, it used to kick me into an asthma attack. So that sort of stuff, that's regulated by the cytokines that's secreted by both T cells and macrophages. So this is considered allergies as a disorder of the immune system. So the immediate allergic response is caused by receptors on mast cells and tissues. Those allergens trigger release of histamine. And I'm sure you guys have probably heard of antihistamine and Benadryl. Those, they knock me out. They make me really sleepy. I'm very sensitive to antihistamine. But they are helpful. That can occur, the histamine, that can occur within seconds of exposure. Symptoms, they vary, with extreme anaphylactic shock, which is a sudden life-threatening drop in blood pressure. You may have heard of that before. Sometimes they're not so bad, but sometimes they're pretty extreme. For me, depending on what the trigger is, my response might not be that bad. Pollen, little sneezing, little coughing. Cats will start out with a little bit of sneezing, some watery eyes. And if I'm, let's say, for example, my aunt has 3 cats, and I go to her house sometimes, I can stand to be in her house for maybe an hour before I'm sneezing and coughing. If I stayed in there all day, I am usually sick for at least a week afterwards if I get out of the house. If I don't, I will just have an asthma attack. If I stay in the house, won't go away. And, asthma attacks sometimes are extremely scary. Sometimes the medicines don't work. So I just prefer not to put myself in those situations. But it's usually a delayed response and a negative one. But it's different for everyone. So you can have delayed allergic responses. Those initiated by memory T cells at the site of an allergen contact in the body. So you can have a skin test for TB. A positive test result is this red hardened skin at the test site, and that'll show previous exposures to TB. You can also have contact dermatitis to poison ivy, jewelry, cosmetics, and so on. So these are things that, you have a cheap ring, it turns your finger green. You brush up against poison ivy and you break out. A lot of makeups, a lot of different kinds of eyeshadow, a lot of different kind of foundations actually really bother my skin too. So I don't wear a lot of those. I'll wear something like a tinted moisturizer because anything cheap, I have a reaction to. Anyway. So autoimmune diseases, like when cytotoxic T cells or antibodies attack the body's own cells, that's what that means. The cause is unknown, but you get something called myasthenia gravis. So that is a neuromuscular junction that doesn't work properly. Then you get weak muscles as a result. MS, multiple sclerosis, the myelin sheaths of the nerve fibers break down, and that causes a variety of neuromuscular disorders. So different things can happen, but function is at stake. Correct function of the muscles. You have systemic lupus erythematosus. That's various symptoms prior to death due to kidney damage. So it's pretty variable. There's not really. I've known one person in my life that has had lupus, and it's kind of all over the place. And then rheumatoid arthritis, the joints are affected. For example, this right here, this is an autoimmune disease. And it's very painful from what I understand. That's on the inside. There's not a lot you can do about it, but maybe you tried to get something to stop the pain. And we're getting close to the end of our lecture this week. So AIDS, that is something that you've probably heard of before over the years, acquired immunodeficiency syndrome. Starts with HIV, which is the human immunodeficiency virus. So it starts with the virus that lives in and destroys those helper T cells that I was talking about. So the body at first is able to maintain adequate T cell numbers for a certain amount of time. But without therapy, that number eventually is going to drop. And then that leaves the person unable to fight off the viruses, fungi, or bacteria that normally, we fight every day. And it's something that I think we take for granted, but outside on a windy day and you get dust blown into your mouth and who knows what bacteria was on the dust, but you just ate it. And that's what happened to me before. That's why I use it as an example. And at the time I was like, thank goodness I have a good immune system. Here I'm like, I'm not worried about it. I have an immune system. Just make sure I stay as clean as possible and, the body does the rest. But it's something we take for granted. So the HIV, that's transmitted, of course, by sexual contact with an infected person, including vaginal or rectal intercourse or oral/genital contact. We know this. Needle sharing or babies born to women who are infected with HIV. Those are the typical spread methods. And as of 2020, an estimated 37.7 million people worldwide were living with HIV. There were 1.5 million new HIV infections in 2020. That's a 23% decline from 2010, which is great. Heterosexual contact and IV drug use, those account for the greatest percentage of increase in new cases. And then total deaths attributed to HIV or AIDS is over 36.3 million. I have a few thoughts that don't really pertain to this, so this is kind of like a side street, if you will, just for a moment. I read this book for my virology class about, I'm trying to think of the name of it. One second. It's called Cured, about people who defeated HIV. It's about two patients who had very positive body responses to certain medicine. But the book is so in detail and in depth that it really can put things into perspective. If you like to read, I love to do this. I throw these books out there. It's a really good one, just talking about the way that it was measured and how the medicines were determined, and then, a follow-up after the fact. The other thing I was going to say is that I remember vaguely, and I'd like to relook into the facts on this, but I found it interesting. During my evolution class, my professor talked about how primates, we know that chimpanzees are known to be the likely source of HIV-1, and then that humans are variably susceptible to the disease. So this is where they think that it came from. But their primates, there's something called SIV, which is simian immunodeficiency virus. And my professor was telling me that they think that primates have lived with this for a long time. So it's something that they've gotten kind of like the common cold. And they've just had it for many, 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 many years. I don't know how many thousands of years. I'd have to do a little research to be accurate. But their genetics and their bodies have kind of adapted to it. But for us as humans, it's a relatively new disease. And so our responses were very extreme, especially in the beginning. Now we're starting to understand it a little better, and I think coming up with better medical advancements and trying to find, different things that work for different people. Anyway, that was a sidestep. Very interesting. Look it up. I always encourage, if I'm wrong about something, I would love to be right. So if I'm wrong, email me, shoot me an article if you found something interesting. I think that stuff's really cool. Here's a visual on that. You guys can take a look in the book. That is on page 517. And so here, the number of deaths have dropped by 39% since 2010. This is what I was saying a second ago, due to the antiviral medication. So they've been making some improvements, and it shows in the percentage numbers. Again, I like stats, so that's good to see. Advances in treatment have reduced the serious complications and really prolonged the life of HIV carriers or people who actually have progressed into AIDS. It has helped. The sooner drug therapies have begun, the better the chances that HIV cannot destroy the immune system. Some of the newer strains, though, are drug resistant. So we've talked about this with bacteria. Resistance can develop. So new AIDS vaccine trials are underway. The RB144 is a vaccine that finished phase 3 trials in 2009. Now, I had to look this up to whenever I first started to teach this class to be sure that I understood. So for most clinical trials, there are 3 phases. Occasionally, there's a phase 0, a 1, 2, and 3, and sometimes a 4, depending on the drug. So what phase 3 means in this case is that it's compared the safety and effectiveness of this new vaccine with the already existing methods, the current standard treatment, because they don't know what is better yet. So they'll pick their participants at random, and try to move forward from there. these are some things that we've gone through. Well, not we in this class, but you've heard over the years. So HIV infection is preventable. 1, to abstain from sexual intercourse or have a long-term monogamous partner. You pick one person to be with who you know is free. You get tested, of course, beforehand and make sure you guys are good to go. Be aware, of course. So knowledge is power. So know that having relations with individuals who use intravenous drugs, it's risky behavior. I'll let you read through all of these. These are things that are important to know. You just need to be aware. That is the bottom line here. And I think this is the last slide that I'm going to go through today. Yeah, sure is. Okay, so I'll stop that chapter here. This was kind of a long lecture, but I think it's very informative, a good overview of the way that our immune system works. As always, if you have any questions, email me, and have a great day.